My name is Georgiana Silvera. I am a master's student at SUNY College of Environmental Science and Forestry. And today I am presenting some of the work that I have been doing with Jacqueline Frere, Jonathan Cohen, and Kristen Schuler, studying anticoagulant rodenticide exposure in fishers in the Northeast US. So first, some introduction. What are anticoagulant rodenticides? They are one of the most common methods of lethal rodent control used across the globe. They work by inhibiting the ability of blood to form clots, and they are so widely used because they are both widely available and require relatively low effort for use. What is the problem with this widespread use of anticoagulant rodenticides? The problem is that widespread exposure has been documented in non-target wildlife, including invertebrates, aquatic species, and most relevant to our work, avian and mammal predators, which has caused a lot of concern around the continued use of these compounds because so many of these predators populations are small or locally threatened, and so there is concern with the continuing use of these compounds. However, while this individual exposure has been documented in individual animals and mortality from rodenticide exposure has been shown, we don't really know how these compounds affect species at the population level. And thus, it's been challenging to develop management or management recommendations or changes to regulation because there really is no alternative for these compounds currently on the market. And so it's hard to introduce bans or restrictions on their use without knowing whether and how they're affecting wildlife populations. The three typical endpoints that we look for a compound's effect on wildlife species are its effects on growth, survival, and reproduction. And we really don't have much data about how rodenticide affects any of these endpoints. They're also pretty challenging and expensive to collect data on these endpoints. And so instead of trying to directly measure the effects of rodenticide on populations, we instead are going to fit a comparison between the rodenticide exposure and population trend in animal populations to try to figure out if the relationship between rodenticide exposure and population decline is strong enough to warrant future research and regulatory action. So this is our main question with this research. When accounting for other drivers of fisher population trend, is rodenticide an important predictor of population decline? As the question says, our main species for this research is the fisher, which was done for a couple of reasons. So fishers are the largest member of the mustelid family in North America. They live across Canada and also on the northwest and northeast coasts of the United States. This project actually grew out of an ongoing study of fisher survival and reproduction in New York State. Radio-collared individuals from this study who were otherwise healthy were actually found dead from relatively minor injuries. And when tested, rodenticides were present in their systems, which sort of started to raise concern for managers in New York State. And we started to collect data on rodenticide exposure in these species. Fortuitously, some other states in the region, particularly Pennsylvania, Vermont, New Hampshire, and New York, were also collecting data on rodenticide exposure in fishers because of similar concerns, and were willing to share their results with us as well, which has really allowed us to perform this huge region-wide analysis of rodenticide exposure in fishers to get a really clear picture of what might be going on with fisher populations in the region. And so obviously the first thing that we have to do to start answering this question is to characterize rodenticide exposure in fishers in the region. And so here is a map showing the location of fishers that were tested for rodenticide in this region. And what's kind of cool about this data set, these 285 tests for rodenticide, is that they really represent more of a snapshot of the current population as opposed to other studies that have investigated rodenticide exposure in wildlife. Most studies are based on rodenticide tests in opportunistically collected carcasses of animals, which might be biased in terms of the numbers and concentrations of rodenticide detected in these species versus these fishers, which were just harvested for regular fur trapping and with no indication of effects of rodenticide exposure. So this really gives us more of an overall picture of rodenticide exposure in this region. All of the rodenticide tests were done at the University of Pennsylvania by Dr. Lisa Murphy. While the concentrations of rodenticide compounds were quantified in these tests when possible, we actually will not be using those concentrations for further analyses. It's really not known how liver rodenticide concentration translates into health effects for species, because the concentrations found in liver are so strongly related to the initial dose of rodenticide that the individual was exposed to, time since exposure, individual metabolism. There's so many factors that go into that, that concentration of rodenticide in the liver that it really doesn't tell us much. So instead, we will be using the presence of at least one rodenticide in the fish or liver, and the number of compounds, the number of rodenticide compounds detected in those livers, as a metric of both extent and proxy of exposure versus those concentrations that just really don't tell us much. And here are some of the results of those tests. So the numbers on the states show the number of individuals with at least one rodenticide compound detected, as well as the percent of total tests from that state with at least one rodenticide compound detected. And the colors of the map show our estimates of numbers of compounds detected in fishers interpolated across the region using Kriging methods. And so we immediately see this really strong sort of geographic pattern in exposure with the highest number of compounds detected in southern Vermont and New Hampshire, and the lowest numbers of compounds detected actually in northern Maine, which really indicates to us something is driving this exposure of fishers across the region. Each cell in this raster that you're looking at is 30 kilometers squared, which is the approximate home range size of male fishers in the region. And we used the values of all of these cells as our metric of predicted rodenticide exposure in our later population models. So now that we have sort of characterized exposure across the region, the next step is to characterize population trend. And to do this, we used harvest records provided by our state partners. And while raw harvest on its own doesn't really tell us very much about population trend, when corrected for effort, harvest has been shown to be a useful index for population trend. And so we just fit simple linear regression lines of catch per unit effort by year to estimate the approximate trend of the population. You'll see two examples on the slides from New Hampshire, but we fit these same regression lines for every management unit across the region. However, as you might have noticed from that initial table that I showed, there are a lot of differences in the ways that these states collect data, the amount of effort data that's available, the number of fishers trapped. And so we're really working with data that's very different between the states and really isn't comparable across states. And so we really didn't feel comfortable just using our raw slope estimates as a metric of population trend. So instead, we fit these models in a Bayesian framework, and we estimated the posterior distribution of our slope estimates for each management unit. We then calculated the percent of the posterior distribution that is less than 0, which gives us the probability that those catch per unit effort slopes are less than 0, and thus the probability that the catch per unit effort is declining over time. And we used that probability of decline estimate as our metric for the propensity that the population of fishers in those management units have for decline. And you will see the results of all of those slope estimates there on this map, with the darker purple management units indicating a higher propensity for population decline in that area because of a higher probability of the catch per unit effort slope that is less than 0. So this is our sort of characterization of fisher population trend across the region. However, to make these estimates comparable to the rodenticide estimates, which were those 30-kilometer square home range squares, we took a weighted average of the propensity for population decline estimates in each of those 30-kilometer square cells where we have a rodenticide exposure estimate from those previous maps. So this will allow us to directly overlay the population decline estimates with the rodenticide exposure estimates and use those corresponding squares to fit a trend of population decline versus rodenticide exposure. And you will see the results of that model on the slide here. We were actually really surprised to see such a strong initial relationship between rodenticide exposure and propensity for decline in these populations. You'll see a positive slope of about 0.2, which again was just a much stronger relationship than we were expecting to see, and potentially indicates to us that the rodenticide exposure maybe is an important driver of fisher population decline in these regions. And this type of result has never really been shown so strongly before in rodenticide. However, this is not the end of the story, because we know that there's more things than just rodenticide, of course, that affect fisher population trend. And so while we have this sort of initial positive relationship between propensity for decline and rodenticide exposure, it may be due to other factors. And so our sort of next steps for this project are to incorporate other known drivers of fisher population trend, such as the availability of nesting and denning sites for raising kits, and the availability of large forested areas, which they need for their large home range sizes. And so we expect to see this relationship change a little bit. It may strengthen as we sort of account for other drivers of population trend. It may sort of fall out of favor as we start to include other metrics of population trend. However, these initial results are really concerning to us and potentially suggest that rodenticides are having a negative effect on fisher at the region level in the northeastern US. I cannot finish this talk without a huge thank you to all of our state and university partners who have really made this research possible. I want to thank all of our colleagues for both advice on using the harvest data and working with the rodenticide data, for actually collecting and analyzing all of these fisher rodenticide concentrations. We would never have been able to collect such a large and extensive data set without the partnership of all of these individuals. And so, yeah, I just want to end off with a huge thank you to all of them for making this work possible. And yeah, thank you so much for listening. And please let me know if you have any questions about the talk. Thank you.